Hi, and thank you for watching my video presentation which will outline the features of algae blooms and the effects that global climate change is having on them. You may be wondering, what exactly is algae? Algae include a diverse group of aquatic phototrophs ranging from microscopic microalgae to macroalgae such as kelp and seaweed. Phototrophs are primary producers that are vital in all ecosystems as they are the foundation of extensive and complex food webs. Through the process of photosynthesis, phototrophs create their own nutrients and produce oxygen that provide energy and nutrients to organisms of higher trophic levels. Algae also demonstrate an ability to fix carbon. Algal blooms refer to large populations of microalgae or cyanobacteria growing at a rapid rate, changing the environmental conditions. These blooms occur naturally but are more frequently triggered by man-made causes. The type of bloom is determined by the type of algae present, as each species have different characteristics determining their level of harm on the environment. Nuisance algal blooms are more of an annoyance than a health risk and refer to blooms of macroalgae, or seaweed. These high biomass blooms interrupt commercial and industrial tasks by polluting beaches, congesting desalination plants, and impairing fisheries. Harmful algal blooms, or HABs on the other hand, may contain toxin-releasing species of algae and severely disrupt the ecosystems they infiltrate. Even if the algae do not produce toxins, the sheer size of the bloom will have devastating effects on the ecosystem. The most common groups of microalgae that form harmful algal blooms are dinoflagellates, diatoms, and cyanobacteria, but generally a bloom will contain predominantly one species of microalgae. These groups have been found to cause algal blooms in both marine and freshwater. Diatoms and dinoflagellates are primarily responsible for the formation of algal blooms in the ocean, with dinoflagellates more prevalent in warmer waters and diatoms frequently cold waters. HABs in the ocean can occur in any region with a coastline. Cyanobacteria have been identified in marine environments but primarily dominate freshwater systems, which are rich in nutrients, encouraging the proliferation of algal blooms. They're commonly called blue-green algae, and although they're not technically algae, show similarities in their photosynthetic properties and bloom consequences. The life cycles of HABs are not fully understood, with very few species having their full cycle studied. However, two clear phases have been identified, a planktonic and a benthic stage. Dinoflagellates, diatoms, and cyanobacteria show similarities in their life cycles, but each species is morphologically and physiologically unique. The planktonic phase occurs in stages where cells are optimized for growth and reproduction. The bloom is initiated when water conditions are favorable, triggering resting cells to emerge from the sediment for germination. Each species have unique conditions that will trigger bloom initiation, and seasonal germination has been expressed in some HAB-causing algae, which ensures the survival of the species regardless of external environmental conditions. The bloom develops as newly emerged algae cells photosynthesize and reproduce, exponentially increasing the population size. When all the nutrients in the water have been exhausted, the blooms stop growth and undergo reproduction, forming resting and temporary cysts. Unlike temporary cysts, which remain in planktonic phase, resting cysts have a mandatory dormancy period in the benthic zone, increasing their chance of survival even under poor environmental conditions. The benthic phase occurs on the sea floor where resting cysts sink into the sediment and become dormant. The cells of some species have been shown to remain dormant for centuries and sustain the ability to germinate. The areas where sediment of cysts settle become dormant reservoirs, which will spawn new blooms when the species-specific environmental conditions are met. There are many different types of harmful algal blooms that can be found all around the world. Red tides are in most cases caused by large dinoflagellate populations and are observed in coastal ocean waters. The color of the tide could appear red, green, or brown, which is determined by the species of microalgae or cyanobacteria present. Red tides with toxin-producing species can cause outbreaks of illnesses such as diuretic shellfish poisoning and amnesic shellfish poisoning. Shellfish are contaminated by toxins produced in harmful algal blooms, and when ingested can result in symptoms ranging from mild discomfort to death in extreme cases. Cyanobacteria are the primary cause of harmful algal blooms in freshwater and brackish estuaries and are often referred to as cyano-HABs. Some cyanobacteria species in these blooms release toxins that can contaminate drinking water, causing serious illness or again death. The physical presence of cyano-HABs also reduces the amount of light penetrating the water column, affecting productivity of local organisms. High cyanobacterial abundance has been linked to higher nutrient concentrations that provide the optimal conditions for cyano-HABs, which becomes a problem when decomposition of dying organisms lowers the oxygen levels in the water to hypoxic levels, consequently killing the remaining aquatic life. Climate change is influencing ocean dynamics and productivity, which consequently affects the dynamics of algal blooms. Sea surface temperatures are rising as a result of global climate change, which is the main factor influencing changes in algal bloom dynamics. Coastal waters are warming faster, which is causing the displacement of marine life on a global scale. When water temperatures rise too high, organisms migrate to new habitats with optimal water temperatures for survival. Algal blooms are susceptible to small shifts in environmental conditions, and increased temperatures in both marine and freshwater environments have been shown to increase the growth rate and abundance of planktonic algal cells. The ocean is also becoming more acidic as it cycles atmospheric carbon. Atmospheric carbon dioxide dissolves in the ocean, resulting in increased acidity, which kills local marine life. High abundance of microalgae has been linked to a decrease in the pH of the water it occupies, which explains why coastal waters are changing more than open waters. With these changing ocean conditions, HABs are expected to increase in severity and frequency in both marine and aquatic environments. Eutrophication occurs when a water system is saturated with nutrients. This is primarily a human-caused process which is increasing the severity and frequency of HABs, and is also accelerated by climate change. Nutrients from fertilizers and wastewater runoff enters water systems, providing an excess of nitrogen and phosphorus, aiding in the proliferation of harmful algal blooms that negatively affect environments and ecosystems. It's been shown that algal blooms can have devastating effects on environments and are becoming more frequent as global climate continues to change. Continuing efforts to understand and monitor blooms will lead to more effective ways to minimize health risks and protect the environment. Safety standards have been introduced in most regions to reduce outbreaks of toxin-related illnesses such as ASP and DSP by monitoring concentrations of algae-producing toxins in harvested seafood. This has proven to be effective at minimizing health risks. Accurately predicting blooms has also allowed preventative action to be taken before damage to environments has been done, and monitoring water systems is a tool to identify potential health and environmental risks. Both are used around the world, and better technology is being developed yearly to assist in the monitoring of blooms. One such development is the use of unmanned drones in monitoring and detecting blooms. Chemical management of blooms have also been explored to find a potential treatment to suppress or kill bloom-causing species. Successful trials have been conducted using hydrogen peroxide in freshwater. A better understanding of the life stages of bloom species would allow for better models to predict future blooms and better chemical treatments to be created. With more time and further exploration of blooms, we may be able to find ways to manage and prevent toxic algae in the future to reduce their harm to the environment